In this video, I'm going to compare and contrast MHC types. So MHC stands for Major Histocompatibility Complex, and there are two different types. There's MHC class 1 and MHC class 2. And MHC complex consists of genes encoding cell surface glycoproteins that are required for antigen presentation to T cells and also responsible for rapid graft rejection. And this means the acceptance or rejection of transplant is controlled by MHCs. So if someone has a transplant, like a kidney transplant or a liver transplant or any kind of transplant, and it is not accepted and it was rejected, that is all up to MHC complexes, but then it's also up to it if it's accepted as well. So accepted or rejected, that's controlled by MHCs. So MHC class 1, it's present on almost every cell in the body, acting to present endogenous antigens that originate from the cytoplasm. These antigens include not only self-proteins, but also foreign proteins produced within the cell, such as viral proteins that take over the cell's machinery in order to replicate the virus. So it takes over the parts of the cell that replicate viruses. When the proteins become degraded, the peptide fragments can be transported to the endoplasmic reticulum, or known as the ER, where they can bind to MHC class 1 proteins before being transported by the Golgi apparatus to the cell surface. So they must bind to MHC class 1 proteins before they can be transported. And once they get to the cell surface, the membrane-bound MHC1 proteins display the antigen for recognition by special immune cells known as cytotoxic T cell lymphocytes. And MHC1 proteins work to present the type of proteins being synthesized within a cell, which can be monitored by killer T cells as part of a surveillance system that identifies and destroys any cells with overabundant or unfamiliar peptide antigens, such as malignant cells, so like cancer cells. It tries to destroy any cells that are overproducing. MHC class 2, they are only present on specialized antigen-presenting immune cells, including macrophages that engulf foreign particles such as bacteria, dendritic cells that present antigen to T cells, and B cells that produce antibodies. So MHC2 proteins present exogenous antigens that originate extracellularly from foreign bodies such as bacteria, so they originate outside of the cell. And when encountering a pathogenic organism, proteins from pathogen can be degraded into small peptide fragments by antigen-presenting cells, which then gather these fragments into the endosome so they can bind to MHC2 proteins before being transported to the cell surface. So again, they must bind to MHC2 proteins before they can be transported. Once at the cell surface, the membrane-bound MHC2 protein displays the antigen for recognition by a different type of T cell, and these would be called the helper T cell lymphocytes. And these helper T cells are activated upon binding to macrophages or dendritic cell MHC2 antigen, causing the release of lymphokines that attract other cells to the area of infection. So those things attract cells to our infections in an attempt to destroy the antigenic material. So the binding of helper T cells to B cell MHC2 antigen stimulates the development of a clone of antibody-producing cells against the antigenic material. So the MHC class 2, they try to attract cells to the site of infection, while MHC class 1 destroys cells with overabundant cells, so things that are overproducing. So that's kind of how they're different. And then basically to compare and contrast these two, MHC1 is present on almost all cells, while MHC2 is only present on antigen-presenting cells. And then MHC1 bind endogenous antigens synthesized in a cell, and MHC2 bind exogenous antigens. So MHC2 is outside of the cell antigens, and then MHC1 is endogenous antigens, so inside of the cell. And then MHC1 present antigens to cytotoxic T cell lymphocytes, and then MHC2 present antigens to helper T cell lymphocytes. So MHC1 is cytotoxic T cell, and then MHC2 is the helper T cell. And then the MHC2 bind the CD4 adhesion molecules on the helper T cells because they present antigen to helper T cells. And then MHC1 bind CD8 adhesion molecules on cytotoxic T cells. So MHC2 uses CD4, and then MHC1 uses CD8. Basically, the makeup of these MHC class 1 and 2. MHC class 1 consists of two non-identical chains, a long alpha chain and a short beta chain, and then MHC class 2 consists of two roughly identical chains, an alpha chain and a beta chain. So MHC class 1 is non-identical chains, and MHC class 2 is a identical chain. So the main function of the MHC class 2 would be to help immune cells in recognizing self cells from non-self cells. And then the helper T cells recognize antigens bound to MHC class 2 proteins. So that's where we get the helper T cells from. And then they're also responsible for rejection of transplanted organs. And then backtrack to the MHC class 1, the major functions are it helps immune system in recognizing self cells from non-self cells, interacts with cytotoxic T cells leading to degradation of antigen. So these are cytotoxic T cells, and class 2 again are helper T cells. And then they are responsible for rejection of transplanted organs. So they're both classes are responsible for acceptance or rejection of transplanted organs. So again, the major differences between these two classes are that MHC class 1 are present in almost all cells, while the MHC2 are only present on specialized antigen-presenting immune cells. And then class 2 is exogenous, and class 1 is endogenous. And then basically, like, the class 2 are the helper T cells, and class 1 are the cytotoxic T cells.